Before we begin our protein synthesis video, we are going to begin to talk about a quick overview of how proteins are made. Protein synthesis begins with the most important factor, DNA, which then will be turned into mRNA through a process called transcription. Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, or mRNA, so it can move the information outside of the nucleus and into the cytoplasm, to the ribosomes. The next step to building a protein involves tRNA, which brings the appropriate amino acids and drops them off to form the protein. This entire section from mRNA to tRNA to amino acids to then building the protein is called translation. Translation is the process where proteins are assembled at the ribosomes. So let's get into the video. First, we will talk about DNA. To begin, the structure of DNA is a double helix. DNA is also made up of nucleic acids, or nucleotides. The nucleotides have three parts. The first part is the deoxyribose, which is the sugar. The second part is the phosphate group, and the third part is the nitrogen bases that I am pointing to here. DNA is located in the nucleus of the cell. Next, we are going to look at transcription. Transcription is changing DNA to mRNA. The first step to transcription deals with an enzyme called the RNA polymerase. This enzyme aids in the process of transcription by unwinding the part of the DNA that is going to code for the particular protein that we are making. Next, the enzyme splits the nitrogen bases that are binded together by breaking the hydrogen bonds that connected them. Now, the RNA polymerase adds the appropriate nitrogen bases to the mRNA single strand. This means that when the nitrogen base on the DNA strand is a T, then the mRNA nitrogen base would be A. And if it was C, then it would be G, and vice versa. The only exception to this rule is if the nitrogen base on the DNA strand is an A. Then instead of having the mRNA nitrogen base be a T, or thymine, it would then change to a U, for uracil, as you can see here. Now, after this entire strand is coded using the particular segment of DNA that we need to make our protein, it becomes the structure mRNA. So as you can see, mRNA is single-stranded, and it begins in the nucleus, but then it can travel outside into the cytoplasm to reach the ribosomes for protein synthesis. Its primary function is to reach these ribosomes so it can deliver the instructions in the DNA code on how to produce a protein. Now comes the process of translation. As we mentioned before, translation is the process of proteins being assembled at the ribosomes. This is also the primary purpose of translation, which is to produce proteins. The first step to the process of translation is the mRNA strand being fed through the ribosome. Next, the tRNA brings the appropriate amino acid and anticodon to the ribosome, and then the anticodon on the tRNA lines up with the appropriate mRNA codon. Then it drops off the appropriate amino acid that it was carrying to build a protein.
The first mutation we'll look at is point. This is where only one nitrogen base is substituted for another. The effect on the protein will not be as severe as a frameshift mutation because it only affects one amino acid.
The next mutation we will talk about is frameshift insertion mutation.
This is when you insert another nitrogen base.
This affects the protein the most because it throws off how each codon is read after the place where the base was inserted. Therefore, the amino acids will be different. For example, instead of this codon being read as ATA, it will be read as AAT.
Next is deletion.
A deletion mutation is the same as an insertion, except instead of adding a base, we delete a base. Again, this will throw off how each codon is read and then will change the amino acids.